Flow Cytometry Center is a centralized resource supporting investigators at NIEHS in their fluorescence-based single-cell analysis studies. Here in the Flow Cytometry Center, we're here to support the investigators' research. They can come to us with studies, things that they're interested in doing, and we can work with them to kind of make things happen for them, to help their science.
It's a great opportunity for research at the Institute that they have the availability to use this kind of technology.
I do joke around that flow cytometry is kind of like magic because you add these antibodies to your cells and you can see them on the screen and you can see where different populations are and what they look like, and I've always been fascinated by that.
There are several advantages in using flow cytometry when studying dynamic biological processes. First, we can measure multiple characteristics of a cell. Second, we have single cell resolution. We examine the cells one cell at a time. But we do this very fast. We can look at thousands of cells in a matter of seconds. We have the ability to isolate or sort a given population of cells. And we can give this population back to the investigator where they can put it in culture or do further biochemical analysis.
For me to be able to phenotype cells, to develop new projects, it's been completely integral and probably always will be.
The Flow Cytometry Center contains state-of-the-art instrumentation, including both sorters and analyzers. Our Fortessa instrument is our main cell analyzer. It has 5 lasers and can do 18 colors.
We have the Melody, which is like small sorter.
Because it's so user-friendly and user-driven, you can easily sort into 96-well plates or 6-well plates.
It's only got 3 lasers. I can only detect 8 colors, but it's much more gentler on cells.
The Melody is great for more basic sorting like GFP or maybe 2-color sorts.
Maria and Donna can set up machine and sort cells, so everything's very easy for users.
We have 2 FACS ARIAs, the difference being the number of lasers that are on there. We have one that has 4 lasers and can do 13 colors, and one that has 5 lasers that can do 15 colors.
The FACS Symphony, which is the newer one, it's called the S6. This one has 4 lasers and can detect 18 colors.
But the unique thing about the FACS Symphony is we can do now 6-way sorting instead of just 4. So we can actually capture 6 different populations of cells simultaneously for the investigators.
Basically used for clinical samples because it's enclosed in a hood to protect the users from exposure to pathogens.
Where I really see flow cytometry going is in the field of mass cytometry, where one can do 30, 40, 50 different characteristics at the single cell, not only in a suspension cells, but also looking at tissue.
The main difference between mass cytometry and conventional cytometry is how the antibodies are labeled and detected. Instead of using fluorescent labels, non-biological metal elements are used to tag the antibodies. These antibodies could then be detected using time-of-flight mass spectrometry. Metal isotopes don't have the spillover issues you see with fluorescent signals. Mass cytometry is a relatively new technology. There's not a lot of these instruments out in the field. I think we're lucky at NIEHS to have this kind of discovery tool that can be used to get big picture insights, which then allow you to dive in to the specifics using the other tools available.
The Flow Cytometry Center as an in-house facility allows investigators to conveniently access our facility.
They are ready to use always, so we can use all instruments even night or weekend.
We usually get contacted by email when a person is interested in sorting. At that point, we set up a meeting. We invite them to come over to see the facilities. We show them around and show them the different instruments and what can be done with each one of them.
If I'm going to be doing sorting, for example, I'll contact Maria to schedule a time, usually a couple months in advance, to make sure that I have availability of times, and then I'll design my experiment to match that.
We have a wealth of expertise of individuals that have been doing cytometry for a long time and can help investigators here in what they want to do.
I've always been fascinated by the capabilities of flow cytometry, and I've always felt like how much you can do with flow cytometry, and I felt like I just kind of scratched the surface.
I have always had the most wonderful time and experience working with the staff doing the experiments. It's always enriched my research and also my day because I enjoy the people so much.